off. So I'm going to talk about how we study sleep and what we're doing in our lab specifically as well. So firstly, let's tell you a little bit about polysomnography, which is how we actually measure all the sleep characteristics to then decide what's what sleep stage and so on. So we have three components to polysomnography. We have EEG, EOG, and EMG, and I know that tells you a lot. So EEG is electroencephalography, and that's essentially we put electrodes on your scalp, and that way we can see the electrical activity from your brain. And as you can see here, there's normally, it looks kind of like a squiggle, but if we use some processing and statistical analyses, we can then actually take apart the signal to see different frequencies, and different frequencies in kind of particular EEG forms are sort of characteristic for specific sleep stages. Then EOG, which is electroencephalography, is we place electrodes around your ear, so your eyes, and that way we can see whether your eyes are moving, whether they're moving up, down, left, right. And this is an example of what the signal looks like. And we also collect electromyography, which is just your muscle activity, your muscle tension. We place electrodes here, and that's more or less what it looks like. Now, why am I telling you about this? I'm telling you about this because we have different sleep stages in our sleep. There's roughly four sleep stages. There's non-REM sleep and REM sleep, and non-REM sleep can be divided into three main stages: N1, N2, and N3. N1 only takes about 5% of the night, N2 takes up the majority, and then deep sleep and REM sleep have around an equal amount of time during your sleep. And as you can see here, these are characterized by different sort of signals in EEG. So when we look at the recording when somebody is asleep, we can then tell whether they're awake in stage N1, N2, N3, or in REM. But if you look closely, you'll notice that stage 1, awake, and REM are all quite similar to each other. And that is exactly why we use electroencephalography, electromyography as well. When you are awake, you obviously move your eyes, your muscle tension is high because you can move around. But when you go to N1, your eye movements start to slow down. They're more sort of rolling eye movements, and your muscle tension falls quite significantly. And then in REM sleep, your eyes move a lot, but your muscle tension is essentially non-existent. So that helps us distinguish the sleep stages. Now, why do we even care about sleep? In our lab specifically, we care about sleep because of memory consolidation. Memory consolidation is this process where the memories from your day are integrated into your memory from your entire life. So the way it happens on the kind of neuroscientific, neural level is that memory stored in hippocampus, which is sort of a temporary memory store, get moved to the cortical layers, so the more kind of outside layers of your brain, to be stored in so-called schemas. Now, what is a schema? This is a schema on an example of a dog. So I'm sure you all know that when a dog barks, it wails, it wags its tail, it has a wet nose, has four legs, right? So you have a sort of collection of ideas about what a dog is or what it isn't. But it can also help you distinguish that both a dachshund and a German Shepherd are both dogs, even though they're quite different. So essentially, when we have memory consolidation, the memories from your entire day get integrated into those schemas so that you can keep this information for longer. And when you sleep, this happens sort of automatically. Random parts of your day will be reactivated as you sleep. But we can also trick the brain to reactivate specific memories, and that is one of the things that we do in the lab. So there is something called targeted memory reactivation, and the way it works is that if you come into the lab and we tell you some information, we make you watch a video, do a task, whatever it is, and we pair a short sound with it, then if we then play that sound later on when you're asleep, then that memory will be reactivated, and depending on when we play it during the night, different things can happen, but in this case, if we play it in deep sleep, then that should lead to improved memory of that event or that video or whatever we showed you. So by reactivating memories that we want to, we can manipulate them in specific ways. And there's a few different applications of that I'm going to tell you about for today, and we're going to focus on one of them later on. So as I mentioned, you can improve memory using memory reactivation, but you can also increase creativity by strengthening links between different kinds of informations, and potentially even improve mental health. And that is what we're going to be focusing on today. Megan?
Thanks very much. So, yes, I'm going to start to talk to you a little bit about sleep and mental health and how they relate to each other now. So, we probably all kind of have an idea that there's this bidirectional relationship between sleep and mental health. If you have a really bad night's sleep, when you wake up, you tend to not feel so great. But also, sleep is really important for emotional processing. And then when we think about mental health, there's also this kind of like relationship between emotional processing and mental health, where if you're not able to process your emotions, your mental well-being is going to suffer. And in many mental health conditions, there are deficits in emotional processing. So, I'm going to talk to you, first of all, about depression and how these all link together in depression specifically. So first of all, thinking about sleep and depression. Again, we probably are all aware that if you're feeling depressed, or people with depression often have a difficult time sleeping. They maybe find it more difficult to get to sleep, or maybe once they do get to sleep, they find that they sleep for longer, or even they might wake up earlier in the morning and not be able to get back to sleep. So there is many different things that can happen with sleep and depression, but also there are differences on the level of the sleep architecture itself. So here we have what we call a hypnogram. So this takes you throughout the night and shows what sleep stage you would be in. And here we have a hypnogram for a normal night's sleep in a healthy person. And here is a hypnogram for somebody who has major depression. And just initially, by looking at them, you can see they look quite different. So, on a regular night's sleep, you'd usually find that when you start to fall asleep, you go through these stages of non-REM into this deep non-REM stage 3, otherwise called slow wave sleep. And you you stay in that slow wave sleep for a little while, and then you cycle through again, but you have more of this slow wave sleep at the beginning of the night, and then we get less and less. And when we look at REM, you can see that actually in the first part of the night, you don't have very much, but that increases as we get further through the night. But in depression, we see that actually we get less time in this deep non-REM stage. So this is a really relatively extreme example where this person has had no slow wave sleep throughout the entire night. Now we also are seeing differences in REM. So first of all, overall, there is a longer REM duration, but also you tend to see that REM occurs much earlier than in the normal night's sleep. And that first REM period is much longer as well. And aside from this, the characteristics that we see in REM are also altered, and we see an increased density of those characteristic rapid eye movements as well. So it seems like sleep and depression are massively interlinked, and sleep is very different in depression to what we would usually see. So now, moving on to depression and emotional processing. So there are many deficits in emotional processing for individuals that have depression. First of all, emotional memory we tend to find that maybe individuals with depression don't remember emotional memories as well, or they might remember them in an augmented way. And then we also have like this negative bias, where you're more drawn towards negative things, as well as a reduced positive effect. So even when you do experience a positive thing, you don't feel it as positively as maybe you would if you weren't experiencing a depressive episode. And then finally, we also have rumination. So this is where you kind of get a thought stuck in your head and you just can't seem to move on from it. And it's like this thought is chasing around in your head and you can't put it away. So, you can see, emotional processing is very much changed in depression. Now moving on to sleep and emotional processing. Again, sleep is really important for emotional processing. If you get a bad night's sleep, you will, and many of you may have experienced this, find that often your mood is very much impacted and you might just not be able to feel very good. And also, you might be much more irritable than usual. Hence the expression, getting up on the wrong side of the bed. But also, again, it has an impact on emotional memory, so you might not be able to remember sort of an emotional event as well as you would have done if you'd had a good night's sleep. And also affective volatility, so you might find that your emotions are more likely to fluctuate than if you had a good night's sleep. So, thinking about how we actually, as a research lab, look at emotions and emotional processing. And we tend to break it down into two different aspects. So first of all, we have valence. So this is like how positive or negative you feel something is. And then we also have arousal. And this is more like how much you feel something. So it could be that there's something that's really, really positive, and you know that it's really, really positive, but it doesn't really affect you, you can't really relate to it, so you don't feel it particularly intensely. Or it could be that there's something that's really, really positive, and it's very personal to you, you relate to it a lot, and you really, really feel that emotion very intensely. So, moving on to what work has been done looking at sleep and emotional processing using this technique that Marta described earlier called targeted memory reactivation. And we found that both REM and non-REM really do seem to have an impact on emotional processing. So when we reactivate memories and emotional stimuli in REM, we find that following this overnight memory reactivation, we get decreased arousal responses to negative stimuli. And then moving on to non-REM, when we reactivate emotional memories in non-REM, we find that we get enhanced emotional memory consolidation, so we remember those memories better. We also have seen this phenomenon called valence updating. So this group paired positive words with a negative picture cue and then used those positive words as a targeted memory reactivation cue during slow wave sleep and non-REM sleep, and found that following this, people actually rated those negative picture cues more positively. And then finally here, reactivating emotional stimuli in non-REM sleep has shown to be able to augment brain activity in the orbital frontal cortex and the amygdala, two areas that have been shown to be linked to depression. And although the reactivation was done in non-REM sleep, this was regulated by REM. So somehow these two different sleep stages are working together for the sort of complete picture of emotional processing. So that's kind of what's already been done, but what are we doing as a lab to try and advance this work? So, a study that I'm working on at the moment is looking at trying to reactivate positive memories during slow wave sleep, so this deep non-REM stage 3. And this is a little bit different to what has usually been done in the past, as I am specifically looking at positive information, whereas most studies in the past have used negative information, mainly because it tends to be more salient. But I've chosen positive information for this because we're hoping that eventually, since the start of the journey, to trying to be able to use targeted memory reactivation for an application in depression. So we don't want to be reactivating people who have depression. We don't want to be reactivating their negative memories. We want to try and improve their symptoms and their mood. So for this, I'm using short positive videos. So these are videos that sort of 10 to 15 seconds long, and they're very well controlled. They've sort of previously been rated as positive by a completely independent source. But also, I'm trying to use autobiographical information. So here, we're asking participants what makes them happy and trying to reactivate that overnight in this non-REM stage 3. And what we're measuring is, first of all, looking at sort of emotional processing. Does this have any impact on arousal and valence? So the morning after and 1 week after, we're asking the participants to rate on a sliding scale for arousal and valence this information that has been reactivated during non-REM stage 3, and we're comparing that to some similar information that wasn't reactivated. But we're also using some more objective measurements as well. So on the morning after the overnight targeted memory reactivation, we are measuring heart rate, which fluctuates in different emotional states, and also galvanic skin response. So this measures the electrical conductivity of the skin, which increases in heightened emotion. So relating it a bit more directly back to depression and trying to see if this might have any impact on depressive symptoms by using questionnaires to sort of measure rumination and see if it has any impact on rumination. Also using questionnaires to assess mood. So some that are quite general mood questionnaires and some that do focus more on depressive symptoms themselves. And finally, as a slightly more objective measure, we're using facial emotion recognition task, where participants are shown a face and they have to try and work out what emotion that face is showing. And this is important because people with depression tend to not be as good at recognizing different emotion in other people's faces, and they also have an attentional bias to negative emotions in this kind of task. So hopefully that's given you a little bit of an idea of what we're doing to try and maybe eventually get targeted memory reactivation to be an application for depression. And now I'm going to hand back to Marta, who's going to tell you a little bit more about PTSD.
Well, hello back, I guess. So, you might be aware that in PTSD people often experience nightmares and insomnia. So obviously nightmares very often link back to the negative experience that somebody has experienced, like the traumatic event. And insomnia is often linked to not wanting to go back to sleep in fear of those nightmares, but also the sleep itself is affected. Some studies show that slow wave sleep, so the deepest part of sleep, has much more arousals in people with PTSD, so it's sort of disturbed. People are more likely to wake up from that stage or move to a lighter sleep stage. But they also might experience night terrors, sleep paralysis, and hypnagogic hallucinations. So there is a lot of evidence that PTSD and sleep are linked together. They affect each other. Okay, but what about it, right? So in 2009, a researcher called Walker has proposed a theory called sleep to forget, sleep to remember, which suggests that our memories are sort of the core of the entire memory sort of file, and we can have a affective tone attached to it. So we have the core memory, which is just facts, and the affective tone, which is the emotional component of the memory. And over time, over multiple nights of sleep, generally that affective tone decreases and the memory is left without the emotional tone. That's the way it should happen and the way it happens in most healthy people. And the reason it happens, it's been suggested that it's because of REM sleep's specific characteristics, which is there is a high level of a neurotransmitter called acetylcholine, which has been linked to memory consolidation processes, but also low levels of neurotransmitters such as noradrenaline, serotonin, GABA, and dopamine. And originally they've been thought to simply cause muscle atonia, which is when you're in REM sleep, you're essentially paralyzed. The only thing of you that moves is your eyes. But it has also been found that the dreams that are present in REM sleep tend to be less negative and more positive. If you experience a negative emotion in REM sleep, it's going to be less intense than a positive emotion you would experience in REM sleep. This has been linked back to the low levels of noradrenaline. Okay? So, why do I talk about noradrenaline in those other weird words? It's because noradrenaline levels are generally increased in people with PTSD. Essentially, they don't decrease as they should when they go into REM sleep. And that leads to disturbed REM sleep, more awakenings from that stage. And this disturbed sleep has been found to be a predictor of PTSD. So if you generally have high noradrenaline levels in your REM sleep, you wake up more, you're more likely to develop PTSD. And this is thought to act through a sort of failure of what we call extinction learning. Extinction learning is essentially you learning that a stimulus is no longer a threat, right? So you had a traumatic event, but it's very unlikely it's going to happen again. It's not a threat anymore, and you don't have to worry about it, right? But that doesn't happen in PTSD. And it's also been found that decreasing that noradrenaline in PTSD patients can actually decrease the amount of nightmares that they have, or the negativity of nightmares. So, as Megan has mentioned, the work we do in the lab is at the very beginning of the journey to hopefully using TMR, targeted memory reactivation, for treatments of depression, PTSD, and so on. So this is an example of the experiment I have conducted and I'm currently analyzing. So in this experiment, I invited some people into the lab. I made them watch short negative videos. So as you might remember, Megan has mentioned that there was a study in the past where people showed participants negative pictures, and then after reactivating half of those pictures overnight, the ones that were reactivated were found to be less arousing in the morning. So I was trying to sort of expand on it and see if more complex memories, like videos, would also be affected the same way. So I showed participants very short, around 10-second long videos, and asked them to rate them for both valence and arousal. Then I checked whether they actually associate the short sound that I gave them with the correct video, and at night I reactivated half of the stimuli, right? So half of the videos were replayed in sleep and half were not. Then in the morning, participants saw screenshots from those videos. They rated the screenshot, then they saw a second screenshot and they had to order them within a video. So essentially it was a memory task. And they repeated that task 2 more times, after 1 week and after 2 weeks. And at the 2-week time point, they also repeated the first video rating task. So that way I can compare how this changes over time, as well as between the acute and non-acute items. So these are preliminary results, and what I mean by that is this is what I found, but I am reanalyzing the data. I cannot tell you that this is definitely significant, so please take it with a grain of salt, but this is what we see in the data. We see that even when we use videos, so the memories are more complex, we still see a reduction in arousal ratings for the items that were cued overnight. We see an increase in a valence rating, so the emotions and memories become less negative, more positive. The higher rating is more positive. And we also see something very interesting, which is that memory is worse for the items that are reactivated. Now, you might remember that earlier on, I told you that if we reactivate memories in non-REM sleep, in deep sleep, then those memories get strengthened. It seems that something opposite happens when we do it in REM sleep. And that just highlights how different the sleep stages are, how different their functions are. I was quite surprised by this finding initially. Then I went back to the literature and I actually found a study who found something similar. So it does seem that it is a fact, it's not a glitch from my study, just good. And I also compared the video ratings from before and after, since the ratings in the meeting were on screenshots, and I thought that might not be a good approximation. And I can see that the change in arousal, so the reduction in arousal, is greater for the items that I cued than the ones I didn't. And also the change in valence, so the increase in valence is higher for the cued items than for the uncued items. And that's it for our little presentation. I'm aware that we went through a lot of information in a very brief time, so if anything's unclear, please feel free to ask questions. We're more than happy to answer them. And thank you for listening.
Are there any questions? Yes.
The slide with the hypnograms on.
Yeah. Go back on.
I suppose a wondering I had was about the influence of drugs, anxiolytics, antidepressants on that picture and what they do.
Yeah, so, I mean, the drugs isn't something that I've looked at massively, but I do believe they do sort of kind of have an effect to normalise it to some extent, but it's not all the way.
So to make it more...
So to make this one look more like this one, but it doesn't necessarily make it look completely like a normal night's sleep.
In fact, I did look at that at one point. That was a bit of time ago, so I don't remember all the details. But it seems to differ quite a bit between different medications. So obviously a medication like, let's say Xanax, right, which is used for anxiety and depression, and is one of the, let's say, heavier medications, will increase some of the slow wave sleep. But that doesn't mean that the quality of that sleep is the same, unfortunately. And some SSRIs, so things like fluoxetine, sertraline, the most commonly prescribed medications, have a bit less of an impact, but they do still change the architecture of the sleep.
Okay, thanks.